think we are good to go. All right. Hello, everyone. Okay, so if you are here, you are expecting to learn about DNA structure. So I started this video a couple seconds ago and it did not record. So that's why we are here. And that is why it says, what is DNA over here? So first and foremost, we need to write our essential question right here, which is going to be, what is the chemical and physical structure of the DNA molecule? So, what is the chemical and physical structure of the DNA molecule? Okay. Oh, it's a question, so we need a question mark. All righty. So our first topic question is, what is DNA? And we are going to write out its full name, which is kind of a hassle to say, and it's a little hard to pronounce, but that is why we say DNA instead of deoxyribonucleic acid. I still stumble on that. So deoxyribonucleic acid. Alrighty, so that's our D, that's our N, and that is our A. Okay, so we can see that the DNA is in our nucleus of the cell, and then they are packed into chromosomes, which has the DNA coiled into it. So each of your body's trillions of cells, trillion cells, except red blood cells, contains about 2 meters of DNA coiled into chromosomes inside the nucleus. So that means that if we were to take a nucleus smaller than this little dot, because we cannot see our cells, whoa, I have stuff all over my hands. Okay, if we were to look at this little cell and all of the DNA inside of it, if we were to stretch it all out, it would be about 6 feet across. Therefore, that means that if you put all of our unraveled DNA in your body end to end, it would go from this earth to the sun hundreds of times. DNA is a molecule. So if we are talking about one of the most notorious molecules, water, or otherwise known as H2O, that means that we have an oxygen atom and 2 tiny little hydrogen atoms attached. So that means that this cluster of atoms is a molecule. So this one's a lot more simple than the DNA molecule. So I'm just reminding us what a molecule is. It is like a book of code that tells your cells what to do. So we were talking about in our cell organelle unit that our DNA in our nucleus tells the endoplasmic reticulum ribosomes what proteins to make and what it needs. So it's basically like an instruction manual for your cells. All right, now we are on to topic question number 2. What are the parts of the DNA molecule? All right, so we are going to now talk about the very, very small and intricate pieces of the DNA molecule. And so first we are going to look at the shape. So we can see that it is this very twisty-turvy pattern here. And it is called a double helix. which is sort of like a twisted. So if we take the spiral of the screw and then we put it into a ladder form, it's like a twisted ladder. Okay. And then when we are talking about the molecule, there are very, very specific building blocks of the molecule. So for water, it is H2O, right? But for the molecule of DNA, we have what we call nucleotides. And so right here, there are 4 different nucleotides that make up DNA. So we have 4 different ones, 1, 2, 3, and 4. So right here, we are going to write nucleotides. That is 1 nucleotide. And we have 3 different very specific, 3 different specific parts of the molecule. So we need to write nucleotide here. Nucleotides, which is just a very, very fancy way of saying the building blocks of the DNA molecule, are composed of, one is going to be a phosphate group. Phosphate. And the first part is going to go right here on this circular portion, which is going to be our phosphate group. And the way that I think of this is, if you have ever heard of pho or pho, it is I believe it's Vietnamese. Yes, Vietnamese soup. And we put the soup in the bowl. So phosphate is going to go in the circular bowl. And then we have our second component, which is going to be here. It is called a deoxyribose sugar molecule. So we're going to put our sugar here. And so we already had dinner, right? We had the soup. And now we want dessert, and that's going to be a very sugary morsel. So we have our phosphate group, then our sugar group. And then we have one of 4 different. So you guys can see we all have a phosphate group here, here, and here. There is a sugar here, 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 and here. But the ending is different for all of these. And that is going to be one of 4 different nitrogen bases. And how we are going to remember this, so we are going to put right here, nitrogen base. And so how we're going to remember that is we had our soup, then we had our sugar dessert, and then it's starting to get really, really late. So it's going to end up being nighttime. So our nitrogen base. So soup, dessert, night. And so there are going to be 4 different nitrogen bases that we are going to choose from, where we have adenine, which is ADENIN. There is also guanine, which is GUANINE. We have our T here, which is our thymine. And we also have our cytosine, which is going to start with a C. Cytosine. So now what we are going to do is you are going to need a couple different colored pencils. We are going to color the phosphate group. So these guys, purple. So we're going to take our purple colored pencils and we're going to color it in. Purple. But we don't have one phosphate group. We have 4 different ones because we have 4 different nitrogen bases. This one's going to be purple. This one's going to be purple. Okay. Then it says to color the sugars yellow. So we're going to color in our little pentagon, yellow, okay, then it says to color adenine red. And we're going to color both of its ring structures red. If you want to color in the little circles, you can color it in red as well. Then we have thymine, which is going to be green. And thymine only has one ring structure. It's a little guy. We can color these in too. Then we have our guanine, which is going to be orange. And then we have our cytosine, which is going to be blue. And that's a one ring structure as well, while the guanine was two. Beautiful. Okay, moving on. So we have our second page, which is where it's going to get a little bit more difficult. And I know that this is a lot of vocabulary, but we're going to take it slow. Okay, so now our third topic question is going to be, how is the double helix structured? How is the double helix structured? And so we already have our phosphate group, which is, remember, our soup. And then we eat our dessert, which is the sugar group. So when we are looking at this double helix, we are going to be considering this outer, the outer poles of our ladder, which are curving. And we are considering this part right here. So all of this purple part. You don't have to do this. I'm just trying to give us a representation. And it's also going to be on the other side. So there's two sides of it. And what makes up these sides are going to be our phosphate and sugar groups. And we consider this kind of like the spine or the structural support for the DNA molecule. And we call this the backbone of the DNA molecule. And so one side, so let's just say the left side, is going to be called the, one side of the ladder, yes, our ladder, which is this guy, is called the 5 prime. So let's say that the left side is called the 5 prime. See, 5 prime. And the other is called the 3 prime here. This indicates how the phosphate and sugar are linked. So we have our phosphate and our sugar. It shows how they are linked, but the two sides of the backbone, so the left and the right, are going to be flip-flopped. So you guys can see that this P and this S is upside down compared to this one, who is right side up. Flip-flopped. Good. Okay. So then we've talked about the outside. Now we are going to talk about this inside part right here, which we see. We have a G and a C, a G and a C, a T and an A and a T. So in between the backbone sides, we are going to talk about the rungs of the ladder. And this is going to be, there are pairs, because you can see that there are 2 in each rung. There are pairs of nitrogenous bases. Yes, bases. Held together by 2 or 3. We are going to say, because these are pairs, these little guys in here are bonds, and we call those hydrogen bonds, because that is what they are made out of. And so whether it be GC or CG or TA or AT, this sequence of base pairs is our genetic code. So that is how we can DNA test, based on the sequence of these base pairs. Now we have our fourth topic question, and we are going to be talking about how do the nitrogen bases pair together. Okay, so these are going to be the most important things that you learn, which is that we have our adenine and our thymine, and they always pair together. A and T always pair together. So adenine and thymine always pair together, and you can count 1, 2, that there are 2 hydrogen bonds. And then we have our G and our C, which is our guanine. Always pairs with our C, which is cytosine, by 1, 2, 3, 3 hydrogen bonds. And a way that we can remember this is we have an apple and then we have a tree. And the apple and the tree always go together. If you want to try to remember the hydrogen bonds with it, you can say that there are 2 apples on the tree. 2 apples on the tree. And then we have our guanine, always pairs with our cytosine for 3 hydrogen bonds. We can say that there is a garage and we are having a car that parks in the garage. So the car goes in the garage. And if we want to remember the 3 hydrogen bonds with it, we have 3 of them because we are living the rich and luxurious life in our garage. Okay, so now we are going to look at something that is going to be a little confusing, but just bear with me. We are going to take a look at our different body styles of our adenine and guanine. So we have 1 ring structure like earlier, and then we have 2 ring structures for guanine and our adenine. So there are 2. And then we have our cytosine, which is only 1 ring structure. And we have our thymine, which is 1 ring structure. So we have certain names for these particular structures. You will notice that this one has 2 and this is paired with 1. Adenine has 2 and it is paired with 1. So we call the pairs that have 2 ring structures, so that's going to be our adenine and guanine, have 2 carbon nitrogen ring structures. So we're going to put our 2 here from 1 and 2. And they are called purines. Purines. Then we also have our 1 ring structure. So thymine and cytosine have 1 carbon nitrogen ring structure and are called pyrimidines. Pyrimidine. That's a lot of I's in one word. Pyrimidines, because it has 1. So we know that 1 always has to match with the 2. So we know that a purine must bond, purine must bond with a pyrimidine. Wow, I am having some issues. Pyrimidine. Wow. If you need to know how to really spell it, it's right there. So we are going to put a rectangle around the purines. So I'm going to choose a different color because I've completely messed this up with my purple. We're going to put a rectangle around our purines, which are our double ring structures. So our A's and our G's, or adenines and guanines. And then we're going to put a circle. You don't have to do the different colors. I'm just doing it so you can see it better. We're going to do our circles around our 1 ring structures, which are called our pyrimidines, and it is our cytosine and our thymine. Okay. Now, And it also says, we don't have to do that. It's okay. All right. So onto the back page where we're almost done. Topic question number 5 is, what are some ways to remember how the bases pair together? So ways to remember. So we have our C and our G, where we could say our car and our garage. And we have our apple on our tree. These will always pair together. Or we could say that the two circular letters paired together while the two stick letters paired together. So obviously our C is a circle and our G is a circle. And then A and T are all made out of straight lines. So those would be our stick letters. Okay, so that is how we know which ones pair together. Now we are going to talk about our hydrogen bonds, which we've already talked about up here. If you want to remember that there are 2 apples and there are 3 cars, that equals the bonds of hydrogen. So, or we can remember that there are 2 angry, that's going to be our adenine. See, our 2 angry tigers. So A and T, 2 angry tigers, and 3, which is our 3 hydrogen bonds. Calm, which is our C for our cytosine. Giraffes, which is our G for our guanine. So 2 angry tigers and 3 calm giraffes. So we know that our adenine and our thymine pair with 2 hydrogen bonds, and then our guanine and cytosine pair with 3 hydrogen bonds. All right, and now we are going to try to remember the purines and the pyrimidines. So we have a big old treasure chest over here. So we're going to try to remember purines. So like we can think of like pure gold. So our purines as our A, which is our adenine, G, which is our guanine, pure as gold. So we can say adenine and guanine are purines. And now we have our pyrimidines. So pyrimidines are, how you spell it is Pyrimides. So we're going to be focusing on the PY. And we know that because adenine and guanine are purines, that means that cytosine and thymine are pyrimidines. So we're going to put cytosine and thymine. So we are going to cut. We're going to cut the pi, which I know that's not how you spell pi, but that's how we're going to remember it. Cut the pi, which is our pyrimidines. All right. So now what you guys are going to do is you're going to fill out the sum it up by yourself, but I'm just going to give you a little clue on some of these. So what does DNA stand for? It's going to be that really long word. Where is the DNA located within a cell? That is going to be on our first page in this section right here. What is the twisted ladder shape of the DNA molecule? That is going to be right here. You're going to look for the ladder and find out what it is called. Then you are going to look at what are the 3 parts of the nucleotide. And that is given right here. 3 parts of the nucleotide. It's also given here as well. What are two nitrogen bases have? What two nitrogen bases have 2 ring structures? So which one has 2 ring structures and are called purines? We can look right back up here at what we wrote. And then which ones are called pyrimidines, which is there. And then what 2 structures make up the backbone of the DNA molecule? We're going to flip over onto our second page, which is the backbone, and it tells you right here. And then what type of bonds hold the base pairs together? We were just talking about that right here. So this is what we are going to talk about right now, which is underneath each letter is a DNA sequence. Write the letter of the base that would pair with it in its complementary strand. So what does A match with? If we have apple, we have a T. Obviously, if we have a T, that means it's going to be A. If it's a G, it's going to be C because a car goes in the garage, and then the car goes in the garage, and you are going to continue this. So this means that when we replicate our DNA, that means that we can unzip it kind of like a zipper, and then we can replicate it based off of, obviously, if it's going to be a T, it's going to be an A. If it's going to be a G, it's going to be a C. So that is how we duplicate our DNA for new cells. One side of the double helix is called the blank prime, while the other is called the other blank prime. Those are 2 numbers that you're going to put there. And then write a summary of this lesson. Okay, I know it says $2 summary. You're just going to write 20 words here that summarize what you just learned. And then you're going to circle if you feel like you got it, if you feel like you kind of get it, and then if you're like, whoa, this is way too hard, go ahead and circle that. But good job, and go ahead and fill that out. And when you are done, please go ahead and turn it into me. I hope you have a good rest of your day. Let me see. We're going to go here. We're going to stop recording. Bye-bye.